Buenas and Hafaday, Manianahus and Maniatluhu, Guahusi Riley, Taitingfong. My name is Riley. I've greeted you in Chamorro, which is the language of the indigenous peoples of the Mariana Islands. My family has ties to the island of Guahan or Guam, which you may or may not know as a tiny island in Micronesia. Small islands will be a big theme of this talk today, the title of which is Oceanic Governance for Biotechnology. So my background is in communication studies, indigenous data sovereignty, and really the heart of my work is about strengthening indigenous governance. So in this talk, I'll be making a case for why indigenous governance is important for biotechnology. And I should also note I'm currently a postdoc at the University of Arizona's Native Nations Institute, and I'm the director of the U.S. Indigenous Data Sovereignty Network and co-director of the newly formed Indigenous Data Alliance. If you have any questions about these orgs, please ask me after the talk. So I'm going to take us through three kind of high-level claims and points today. First, I want to offer some brief background to kind of frame why indigenous peoples are important rights holders, not just stakeholders in biotechnology governance. Then I'll spend the majority of the talk walking you through a case study on gene drive technologies, which was the focus of my dissertation research. And then in closing, I'll just briefly offer a few recommendations or directions for what I'm calling oceanic governance of biotech. All right, let's jump in. So first is some background. Why should we even be thinking about indigenous peoples in biotech governance? Well, indigenous peoples globally make up just under 6% of the population, yet their lands comprise some 1/4 of Earth's surface, and those lands comprise about 80% of the planet's remaining biodiversity. Indigenous peoples have, for millennia, stewarded sophisticated knowledge systems that have allowed them to sustain their lands in these ecosystems. And we've increasingly seen the sciences taking interest in these knowledges, what some may call traditional ecological knowledges. However, scientific research, sort of incorporation of indigenous knowledges, is not always done in an ethical manner. This work comes from my colleague, Dr. Dominique David Chavez, who conducted a global systematic review of 20 years of climate research. And what she found was that the vast majority, 87%, of climate studies were extractive, meaning they engaged or used indigenous knowledges in various ways, but did not seek consent from the knowledge holders, those coming from the communities to whom those knowledges belong, or did very little to no consultation with those communities about how that knowledge was used in the research. Now, this isn't only poor research practice, this is also a violation of indigenous peoples' rights as sovereign peoples. These sovereign rights are enshrined and codified in a number of local, regional, national, and international mechanisms, one of these being the United Nations Declaration on the Rights of Indigenous Peoples, or UNDRIP. And I've summarized just a handful of what I see as relevant UNDRIP articles for biotechnology governance. These cover indigenous peoples' inherent rights to self-determination, their rights to participate in decision-making processes, their rights to free prior informed consent, or FPIC. So in summary, when a biotechnology will have implications for indigenous territories, resources, ecosystems, indigenous peoples really are key rights holders in informing and shaping those decisions. Okay, so now that we've walked through a bit of background, I'll take you into this case study on gene drive. For those unfamiliar, gene drives are techniques of genetic modification that push or drive genetic modifications through populations of sexually reproducing organisms. Let's take the example of a gene drive mosquito, which is a very common target of gene drive. If a genetically modified mosquito mates with a wild type, typical Mendelian inheritance means that just 50% of those offspring will inherit that modification and associated traits. Alternatively, gene drive works to push those changes and the associated drive through nearly 100% of offspring. This has really exciting potential applications in many fields spanning public health, agriculture, and conservation, where much of my work has focused. Gene drives could be used to spread changes that can essentially suppress or crash populations of mosquitoes that spread disease to humans, like malaria and Zika. Similarly, they could be used to suppress populations of pests that spread crop disease. And in conservation, they could be used to target invasive species like rodents or mosquitoes. Where my work has focused in Hawaii, there's interest in using gene drive as an intervention to avian malaria, so addressing the spread of disease, in particular by the Culex mosquito, to endemic endangered birds. In a sort of different example, there could also be gene drive applications for spreading resilience through populations of organisms. There's talk, for instance, of editing coral reef to be resilient to warming and acidifying waters. Of course, a powerful technology like this comes with no shortage of profound ethical issues. These run the gamut from things like public acceptance to regulatory issues, transboundary regulatory issues, some of which have been talked about eloquently by others today so far. Also, uncertainties around downstream impacts on ecosystems, issues of dual use. In the wrong hands, could a gene drive be weaponized in some fashion? And of course, all of these issues really are intensified by the fact that some gene drives are designed to endlessly self-propagate after release into the environment. So in theory, with even the release of just a few of these gene drive carrying organisms with self-propagating mechanisms, we could see large, potentially global, potentially irreversible changes to the environment. And so this raises this understandable concern around containment, and this is where islands then come into the story. So looking to the gene drive literature, there's often, explicit call for the first outdoor field trials of gene drives to occur on geographically isolated islands. You see landmark reports from organizations like the World Health Org, the National Academies of Sciences, Engineering, and Medicine's Gene Drive on the Horizons report, which call for those trials to occur on islands, which they frame as, quote, ideal testing locations for genetically modified mosquitoes, especially those carrying a drive, or as the NAS report says, constitutes an ideal, geographically isolated, contained setting to physically limit the dispersal of organisms. Now, this is, of course, not the only time we have seen this logic that islands' isolation make them ideal test sites for emerging technologies. In fact, this was the very logic that was used to justify hundreds of tests of nuclear weapons throughout Oceania and elsewhere. So this is what we're looking at here, a map of nuclear explosions around the globe since 1945. I added the circle to indicate the region that is Oceania. And while folks tend to be very familiar with the nuclear legacies of places like Chernobyl and Hiroshima, far fewer are aware that the US actually tested its most powerful nuclear weapon ever in Micronesia, in the Marshall Islands, with the 1954 Castle Bravo shot. This explosion was estimated to have yielded the force of 1,000 times Hiroshima. And if the interest in testing these weapons in isolated islands was to contain their impacts, this was certainly not the case, neither geographically nor temporally. Marshallese and their island neighbors at nearby atolls have faced myriad health issues and continue to today, from radio-induced cancers to elevated thyroid disease to reproductive issues. Not to mention, many Marshallese and islanders from this region cannot return home to some of their home islands that are deemed unsafe for habitation due to lingering radiation. So considering this history, seeing the regularity with which these claims that islands make ideal test sites for gene drive really circulated in the discourse, my concern sort of grew, and this really motivated my dissertation research to focus on gene drive and specifically indigenous governance in gene drive. So I took two lines of inquiry and methodological approaches to my study. First was a document analysis. This was really looking to some of those landmark reports on gene drive to understand how does this kind of claim around island isolation emerge in the literature, how is it articulated, what evidence accompanies it. And then secondarily, I engaged in ethnographic research to understand how is engagement happening on the ground in actual island locales where gene drive is being considered as a potential conservation intervention. And so I'm just gonna really scratch the surface of the findings from this research, and I'll start with the document analysis. So Again, I looked to kind of landmark reports, policy recommendations, scholarly articles about gene drive, and I looked for this conversation around island isolation. And what I found was that in the phased pathway recommendations, islands are typically talked about between phase 1 and phase 2, moving from those contained lab settings to physically or ecologically confined field trials. And in this literature, the language of containment and confinement abounds. Containment usually referring to the physical containment of gene drive carrying organisms in the built infrastructure of the lab. Confinement referring to ecological confinement, so geographic, spatial, and/or climatic isolation to limit those drive carrying organisms. And what I found is that when containment is discussed, right, in the lab, this is a highly operationalized, thoroughly detailed process, right? There are many stringent protocols around who can enter a lab where gene drives are being developed. There are layers upon layers of containers meant to limit those organisms to the physical space of the lab. Alternatively, when ecological confinement is discussed, the conversation typically moves to islands, and the measures that would be taken to confine those organisms to that isolated island were barely operationalized, much less thoroughly detailed in the literature, right? Almost as if the isolation of the island will itself perform the task of containment absent the physical infrastructure of the laboratory. Now, again, these are very powerful technologies with potentially global implications, so we would hope that the measures taken to perform this ecological confinement on islands would kind of have more care and attention taken to it. And there was some mention in the 2014 WHO report in which it was noted that if the genetically modified mosquito in this case is a rapidly self-limiting one, separation by, quote, 2 kilometers will probably be sufficient. And this is quoting a 1997 study, and what I understand is sort of common knowledge that mosquitoes really just can't fly that far, right? But of course we know there's other issues, other reasons that mosquitoes spread far outside of their native ranges, human-assisted migration being one of those. And in fact, this is how mosquitoes got to the most isolated archipelago on the planet with the Hawaiian island chain. And so these questions are really important because this recommendation around islands for these initial gene drive trials are not just in the realm of the theoretical, right? These recommendations are informing real on-the-ground practice. This is a screenshot from the UC Malaria Initiative's website, their criteria for site selection, where they work in islands off the coast of various African countries, notes geographic isolation as one of the most important criteria for their site selection. Okay, now I'll just jump through my ethnographic methods briefly. So again, my interest here was in understanding in Hawaii, where gene drive is being considered as one potential intervention for avian malaria, how are Hawaiian folks being engaged and how would they like to be engaged? I should note, and I think folks in here are very familiar, that I would say there's really consensus in the literature on gene drive that some form of community engagement is, in fact, requisite to the release of a gene drive in the environment. And in fact, you get that report from the National Academies of Sciences and these major fora, like the Convention on Biological Diversity, calling for free, prior, informed consent, invoking that UNDRIP language, talking even about the importance of indigenous knowledges to this decision-making process. So first what I did is I interviewed Kanaka Oiwi, or Native Hawaiians, just to understand what is this conversation around gene drive really prompt for them. And so what I found was that Native Hawaiians very frequently reflected on various instances of violation to their self-determination and sovereignty when we talked about gene drive, right? It brought up, memories of the decades-long bombing of the island of Kaho'olawe. It brought up exercise RIMPAC, which is a large warfare military training exercise that happens in Hawaii every few years. It brought up the proposed construction of a giant 30-meter telescope atop the sacred Mauna Kea. And it brought up the memories of the University of Hawaii attempting to genetically modify kalo or taro, which is a culturally sacred Hawaiian plant back in the early 2000s. And so I want to be clear that I do not read these insights as sort of anti-science, right? Rather, I think what this does point to is that for peoples whom regard the land as an ancestor, or as Nolani Goodyear Ka'apua, cogently puts, that the aina, or the land, is not something, but someone, right? And so when land is regarded as an ancestor, the stakes for technologies that can impact the environment are elevated, right? So keeping this complexity in mind, I want to ask you to imagine, maybe close your eyes and picture, what might it look like to design engagement or public conversation that can really create space to surface some of these tensions, to address them, and then to collectively navigate these decisions around gene drive. So keep that picture in mind, open your eyes. When I observed a handful of events in Hawaii, what they actually ended up looking like was something more like this. This is an example of a client viewing room. This is a common practice in marketing, research, and communications. And what I observed was sort of conservationists from various agencies sat on one side of the room, looking through a one-way mirror into the other room where local residents were sat and participating in a facilitated focus group with a market researcher. And in this case, the conversation was much more designed around sort of polling and assessing local awareness and knowledge and opinions about invasive and native species issues, and even advertising, or I guess getting feedback on potential advertisements, so like radio commercials about these vector control technologies for mosquitoes, including gene drive, though interestingly, the word or phrase gene drive was never mentioned in the marketing script. So I don't know about you, but this is certainly not what I have in mind when I think about, the kind of participatory engagement called for by some of these landmark reports, right, in which residents would be treated as equal decision-making partners, or treated as though they have really valuable knowledge and insights to inform decisions about the deployment of gene drive in their home communities. And so taken together, sort of some of the takeaways from my dissertation work and case study are here. I argue that safe and ethical gene drive science engagement, or whatever we think that is sort of limited by the reliance on these frames of containment and isolation. And so this is both in the instance of framing islands as naturally contained and therefore safe trial sites, right? And I argue this obscures the displacement of risk onto those island geographies. And I also argue that strategic approaches to engagement, like the one that we saw in the last slide, actually function in of themselves as an act of containment, anticipating and containing potential opposition, and unfortunately foreclosing opportunities for mutual learning and sharing of expertise. So I'm going to just quickly wrap up here with this third piece, which is some recommendations for what I'm calling for as an alternative to these reliance on these frames, what I'm calling oceanic governance. So I think if we can take a page out of the book of islanders who have for many, many years been expertly navigating the ocean, right, who understand that the ocean does not disconnect our islands, but rather is a pathway for connectivity and mobility. Micronesians, right, navigated some 4/5 of the southern hemisphere using only traditional methods. These are people who understand connectivity quite well, right? So I think if we can take a page out of that book and inform our approaches to governance for technologies that are actually designed to spread, right, instead of this emphasis on isolation, it might look like foregrounding connectivity, valuing relationships, recognizing the interconnectedness of ecosystems, taking a more expansive, not exclusionary approach to what constitutes relevant and valuable expertise toward these decisions, and importantly, engaging history as a vital resource for navigating shared futures. And I have to shout out to the great Epeli Hau'ofa, who's on this slide, who says we should not be defined as peoples in Oceania by the smallness of our islands, but the greatness of our oceans. And I'm going to offer in closing just a handful of resources to point us toward these recommendations. So some literature that might help us reframe isolation instead for connectivity, looking to folks like Epeli Hau'ofa and other indigenous and oceanic scholars who write very eloquently about this. Also texts like this interesting paper from Philosophy and Technology, which is rethinking containment, and some of my own work, again, problematizing this frame of islands as labs. I also think there are many lessons we can learn, in particular from the indigenous data sovereignty movement, about how we might bridge local authority with global governance. Natalie Koeffler has also written about this in Science, and I worked with many colleagues on a paper in Nature Biotech around a global gene drive project registry, including with some people in this room. Very last slide. Also important, as I said, to engage diverse knowledges in these decisions. I wrote about this with my colleague Anika Ula in the Hastings Center report, and I work with Natalie Koeffler, my good friend and co-conspirator in these issues. We're actually working on a book project to address some of these challenges and tensions and how to govern these technologies better. So that is forthcoming. With that, I'll close and say a big Sainama Ase. All the names have disappeared from this slide somehow. But thank you to everyone. Thank you to the planning team for this workshop. And thank you all for the space to share today. I'll take your questions.